Hello and welcome dear students. Today we are going to discuss about the topic genetic drift, that is the bottleneck and founder effect. The main objectives of today's lecture are to know the basics of genetic drift, to understand the relation between genetic drift and evolution, to understand the role of genetic drift in small population, to know the examples of genetic drift, that is the bottleneck and founder effect, and to know the experimental evidence that demonstrates the role of genetic drift and to explain types of genetic drift. Also, we will be looking to understand the difference between genetic drift and gene flow. Dear students, let's start with the introduction to genetic drift, also called as bottleneck effect. It is a change in the gene pool of a small population that takes place strictly by chance. Genetic drift is a mechanism of evolution in which allele frequencies of a population change over generations due to change, that is sampling error. Genetic drift can result in genetic traits or characters being lost from a population or becoming widespread in a population without respect to the survival or reproductive value of the alleles involved. These changes are non-directional, that is, they are not under the influence of natural selection. It occurs due to an error in selecting the alleles for the next generation from the gene pool of the current generation. Drift is a binomial sampling error of the gene pool. What this means is that alleles that form the next generation's gene pool are the sample of the alleles from the current generation. As a random statistical effect, genetic drift can occur only in small isolated populations in which the gene pool is small enough that chance events can change its makeup substantially. In larger populations, genetic drift is slower. Any specific allele is carried by so many individuals that it is almost certain to be transmitted by some of them unless it is biologically unfavorable. In larger populations, the allele frequency of the genes remain relatively stable because the genes are not affecting fitness and do not have a natural selection pressure against the alleles. Genetic drift is based on the fact that a subsample, that is small isolated population that is derived from a larger sample set, a large population is not necessarily representative of the larger set. As might be expected, the smaller the population, the greater the chance of sampling error or misrepresentation of the larger population, and hence the significant levels of drift in any generation. In extreme cases, drift over the generations can result in the complete loss of one allele in an allele pair. The remaining allele is then said to be fixed. Dear students, let's talk about the theory of genetic drift. This theory was given by Sewell Wright in 1930s. The salient features of this theory are that genetic drift is an evolutionary force operating in small populations. Genetic frequency in small population changes by chance because of sampling errors. In small populations, some genes may be lost or reduced and others may increase or sheer chance irrespective of their selective advantage or disadvantage. By genetic drift, a new mutation arising in small populations may either get fixed or lost irrespective of its adaptive value. Genetic drift may fix some non-adaptive traits in small populations. Genetic drifts tend to preserve or eliminate genes without distinction. Isolated small populations of a larger population possess some unusual characteristics or traits not shown by larger parental population. Dear students, let's understand the causes of the genetic drift. Genetic drift usually occurs in small populations, and in small populations with many alleles, any of the allele can become extinct. During genetic drift, there is a change in the frequency of alleles of the population resulting from random variation in the distribution of alleles from one generation to the next generation. In a population with many organisms, there is less chance of losing an entire allele. This is because many organisms contain the alleles and all the alleles cannot be wiped away in one go. If the allele affects the organism such that it causes more reproduction of DNA, the allele frequency increases. If the allele harms the organism, the allele frequency decreases. When the allele frequency increases or decreases because of its pressure in some random organism that survived, it is known as genetic drift. The effects of genetic drift are more significant in small populations. Genetic drift can lead to the loss of genetic variations within the population and therefore can cause harmful alleles to become fixed. Genetic drift can have major effects when a population is sharply reduced in size by a natural disaster, that is bottleneck effect, or when a small group splits off from the main population to found a colony or founder effect. Even though genetic drift is the result of chance, it is affected by many factors such as the number of individuals within the population, where the effect of genetic drift is more predominant in small populations. Individuals die due to random chance. The second factor is the number of individuals contributing in genetic drift, since some individuals do not produce offsprings at all. Another factor is the occurrence of natural disasters that affects the population size. Accordingly, these disasters will increase the significance of genetic drift among the population as well as the change in natural gene flow patterns. Habitat fragmentation is one of the main factors that influence gene drifting, where humans live in non-urban habitats, so non-human populations are dispersed or even eliminated. As a result, gene flow among these populations is decreased while gene drifting becomes more significant, as is represented in the figure. Dear students, let's now discuss about the genetic drift consequences. Genetic drift results in loss of diversity in the genetic pool, where fixation of certain genes may be similar to its initial frequency in the population. Another consequence of genetic drift is the increase in differentiation among populations, where the population of the same species may have different alleles due to genetic drift. This occurs if the frequency of one allele is fixed in a population, while its frequency decreases in another population, since the frequencies of alleles in the genetic pool proceed in different directions. Sampling error can cause more change in allele frequencies of small populations than the larger ones. In small populations, alleles are fixed more rapidly than the large populations, since the gene pool of large population tends to be more stable. However, large populations are affected by genetic drift since they become small as a result of a natural disaster that decreases the population size greatly until the conditions improve to allow the regrowth of the population, as is evident in case of bottleneck effect, or when a small population leaves the group to establish a new colony when the population is threatened, as is evident in case of founder effect. In order to protect a small population from extinction, genetic diversity should be maintained. Consequently, genetic drift should be minimized and mutation should be maintained to facilitate adaptation. Dear students, let's now discuss about the role of genetic drift in small populations. The genetic drift in small isolated population leads to variations or change among population or deems by fixing or eliminates certain alleles. Fixation or elimination of gene mutations or fixation of unfavorable, neutral or favorable characteristics in populations. Establishing reproductive isolation between different deems of a large population and origin of new species. Dear students, let's now discuss about the role of genetic drift in evolution. Genetic drift contributes to the increase or decrease of a particular allele in each population. Therefore, the effect of genetic drift is neglected over the long run in normal populations. However, the effect of genetic drift cannot be neglected if an allele frequency has reached zero unless a mutation produces this allele again. Genetic drift is potentially important in evolution since it determines the fate of a mutation to take place. It determines whether it will disappear or become fixed in a population after a few generations. For non-ideal populations smaller in size, the genetic drift is important even for the common genes. Normally, if an allele is fixed in one generation, it is most likely to increase in the upcoming generations. However, in terms of genetic drifting, what occurs in one generation does not necessarily occur in the forthcoming generation. So if one allele increases in one generation, it may possibly increase or decrease in the next generations. A subdivided population adaptation is a process consisting of two phases. The first phase is genetic drift, where the loss or fixation of some alleles randomly occurs by chance, which in turn helps the population to explore new genes. The second phase is characterized by natural selection of the most beneficial gene that were introduced in phase 1. These genes are exported to other populations by migration. The genetic drift theory has a potential role in the evolutionary process of individuals, wherein the balance between mutation and gene drifting creates a state in genetic variation. Since mutation introduces new alleles, while gene drift may eliminate or fix the new alleles. Dear students, let's now talk about the experimental evidence to demonstrate the role of genetic drift. Maxime Lamotte in the 1950s studied the frequency of banded and bandless land snail. He grouped colonies according to the size or the number of individuals in the population. The small population was designated with 500 to 1000, while the intermediate population was designated, or colony was designated, 1000 to 3000, and the large colony was one with over 3000 individuals. Gene B, that means gene with capital letter B represented, was considered bandless, and the gene B represented by small letter b was considered banded. In large populations, the frequency of the gene capital B is 1 to 3%, while as in small population, the frequency of gene capital B is 1 to 100%. This indicates that in some population, one allele is fixed and its alternate allele is fixed in another population or colony. Dear students, let's now understand genetic drift with some examples. Genetic drift can be observed in the following examples. The American bison was once hunted to such an extent that it became endangered. The population which have recovered today show very few genetic variations. Consider a population of rabbits with brown fur and white fur. White fur being the dominant allele, due to genetic drift, only the brown population might remain with all the white ones eliminated. A couple with brown and blue eyes has children with brown or blue eyes. Even if there is a 50% chance of having blue eyes, brown eyes being the dominant allele, all the children might have brown eyes in the future generations as a matter of chance. A bird has an allele for two different sizes of beaks. Genetic drift might eliminate one of the beak sizes from the population, thus reducing the genetic variation of the gene pool of birds. Hypothesize a plant that produces blue or yellow flowers. If the yellow flowers are destroyed in a fire and the blue allele is the dominant one, the plant will produce only blue flowers. Dear students, let's now discuss the types of genetic drift. Let's first discuss about the bottleneck effect. Yearly or seasonal phenomena of cyclic fluctuation in population density causing periodic squeezing of some of the gene in a gene pool in random fashion is called bottleneck phenomena. In the bottleneck effect, the population size severely decreases due to competition, predators, or diseases. The frequency of certain alleles in a population changes because the organisms that carry them are eliminated. The others increase in number because they are the only alleles left in that population. This phenomena is observed during natural disasters like droughts, volcanoes, eruptions, earthquakes, overhunting, forest fires, human activities, etc., and leading to the death of most of the population, as northern elephant seals have reduced genetic variation, probably because of a population bottleneck humans inflicted on them in the 1890s. Hunting reduced their population size to a few as 20 individuals at the end of the 19th century. Their population has since rebounded to over 30,000, but their genes still carry the marks of this bottleneck effect. They have much less genetic variation than a population of southern elephant seals that was not so intensely hunted. A bottle is filled with marbles where the marbles represent the individuals in a population. If a bottleneck event occurs, a small random assortment of individuals survive the event and pass through the bottleneck and into the cup, while the vast majority of the population is killed off remains in the bottle. The genetic composition of the random survivors is now the genetic composition of the entire population. Dear students, let's see how the bottleneck illustration can be applied here in this figure. In the original population, there are green, red, and orange individuals living in a habitat. Orange makes up the majority of the population. The colors represent alleles for a color gene. When a disaster strikes, there only a few random individuals make it out alive. As you can see, by pure luck, most of the survivors are red with a few green and just one orange. Over the time, the population grows again, but now majority of the members are red and there are just a few orange ones. This is very different from the original population before the disaster took place. This illustrates that when a bottleneck event occurs, the variety in a population can decrease and the proportions of different alleles can change completely. As illustrated in the figure, during the bottleneck event, the parent population has significant genetic variation, while as post the bottleneck event, only the green and yellow alleles remain there. When a population is small, genetic drift reduces genetic variations faster. So alleles could even get eliminated from the population. Dear students, let's now discuss about the founder effect. When a few individuals or a small group of individuals from some large population invades a new or isolated geographical region, they become founders or founder members. The unique frequency of genes that arise in a population derived from small bands of colonizers or founders has been called the founder effect or founder principle. This principle was proposed by Harvard evolutionist Ernest Mayr in 1940s. These founders carry only a limited portion of parental gene pool. Their gene pool may contain certain alleles in a very low frequency or may lack a few alleles. In the founder effect, a new population is founded in a new location due to physical or geographical barriers. The new population formed does not interact and mate with the original population. So the new colony is isolated from the original population and the founding individuals may not represent the full genetic diversity of the original population. As a result, the allelic frequencies of the new population will be different from the original population. Dear students, let's assume an individual's shape is determined by its alleles for a particular gene. So random groups that depart to establish new colonies are likely to contain different frequencies of squares and circles than the original population. So the allele frequencies in the colonies' small circles may be different relative to the original population. Also, the small size of the new colonies means they will experience strong genetic drift for generations. The key difference between the bottleneck effect and the founder effect is that the founder effect involves migration. A small group of individuals separates from a large population and establishes a colony in a new location. For example, a few individuals may migrate from a larger continental population and become the founders of an island population. The founder effect has two important consequences. First, the founding population is expected to have less genetic variation than the original population from which it was derived. Second, as a matter of chance, the allele frequencies in the founding population may differ markedly from those of the original population from which it has isolated and moved out. Dear students, let's now understand the difference between genetic drift and gene flow. Gene flow is the movement of genes between population, species, or organisms. For example, bacteria can transfer genes between different cells. On the contrary, genetic drift refers to the random selection of a gene in a population when individuals from one population migrate to some other population and breed there. Gene flow occurs. So gene flow changes the allele frequencies that result to increase a particular gene. So gene drift is counteracted by gene flow since a population does not usually stay small for a long period of time to be influenced by genetic drift. Gene flow can result in an increase in the genetic variations. So gene flow may occur as a result of the passive seed dispersal or active migration between habitats. Studies have shown that only one migrant per generation can increase the genetic differentiation among a population. It can also prevent the genetic drifting effect in decreasing the genetic variation among populations. Note that this rule applies only for ideal populations, while the non-ideal populations may require more than one migrant to counteract the genetic drift effect. Gene flow and genetic drift are two processes that limit a population's genetic diversity and lead to evolution by variations. Gene flow is the exchange of genes between two populations as is depicted in the figure. The development of a new species is enabled via gene transfer. So gene flow is mediated by reproduction and vertical gene transmission from parent to the offspring and may occur between the two populations of the same species. While as genetic drift is an evolutionary process in which a population's allele frequencies shift over generations or pure chances, the gene flow is generated by organisms migrating into the new populations, while as genetic drift is typically caused by population decline or the creation of a new population. On the contrary, the fluctuation of allele frequencies in a small population is known as genetic drift. Alleles with high frequencies may become prominent in a population due to genetic drift. So the influence of each occurrence on a population's allele is the key distinction between gene flow and genetic drift. Dear students, let's now summarize the lecture. Although genetic drift happens in populations of all sizes, its effects tend to be stronger in small populations. The effect of genetic drift is negligible in large populations, but in small breeding populations, all the limited number of progeny might be the same type with respect to certain gene pairs because of chance alone. So genetic drift drives relatively more alleles towards extinction for smaller populations as expected, whereas it introduces slightly deleterious effects for larger populations. Also, the heterozygotes condition present in ancestral forms finally gets converted into homozygous form, leading to the loss of an allele. When small population of a species are isolated out of pure chance, the few individuals who carry certain relatively rare genes may fail to transmit them. The genes may therefore disappear and their loss may lead to the emergence of new species, although natural selection has played no part in the process. Genetic drift affects the genetic makeup of a population, and new population is genetically distinct from the original population, hence it plays a role in the evolution of new species. Genetic drift generally does not produce a fit between organisms and environment and can in fact result in non-adaptive or maladaptive changes. Because of the genetic drift, allele frequency changes occur regardless of those allele carrying individuals and their fitness. Unlike natural selection, genetic drift does not depend on an allele beneficial or harmful effects. Instead, drift changes allele frequencies purely by chance as random subsets of individuals, and the gametes of those individuals are sampled to produce the next generation. In the end, regardless of the population size, genetic drift equals to the allele loss or fixation. So dear students, unlike natural selection, genetic drift outcomes are unpredictable. It is like flipping a coin without knowing which face you will get. Gene drifting outcomes are always due to pure chances. Dear students, it was all about today's lecture regarding genetic drift, that is bottleneck and founder effect. Hope you have understood it well. See you next time with a new topic. Till then, take care. Goodbye.